So now we've gotten our genes replicated, transcribed into mRNA, spliced them, capped them, polyadenylated them, and now we need to make protein. And so today we're going to talk about translation, the process of protein synthesis, wrapped around the deadly poison ricin. To get to ricin, we need to talk about castor beans and castor oil. What are castor beans? They come from the castor plant. Image of a castor plant is shown on the right here. When these plants flower and create seeds, these are the castor beans inside these little red things here. And if anyone has watched like the old Looney Tunes cartoons and things, you see like the baby get shoved castor oil in their mouth. It was used commercially as a laxative or like form of punishment for little kids way back when you could do those things. Not anymore. It's still used for ingredients of, you see, plastics, other odds and ends. But the reason why we talk about castor oil is that the handling of the beans is a simple extraction process where you take the oil out, and then there's a leftover water-soluble part, which is where this compound ricin resides. And it is cytotoxic. We'll talk in detail how. The reason why you have this tight association with ricin, which can exist in other plants, but the reason why it's tightly associated with the castor plant is that these plants, and the bean particularly, makes a massive amount of this biotoxin per unit weight of the bean. Symptoms of ricin poisoning are listed here. There's a long onset. It's multiple days. You can see abdominal pain, vomiting, a lot of gastrointestinal distresses, and it really has a systemic influence on the individual, and you'll understand more about that when we talk about its mechanism of action. The one other thing to add on this is that oil and water-soluble extraction is extremely easy to do, and this is the reason why you may see instances of ricin threats or ricin terrorism. There was, in fact, one, I think, about a month ago where castor beans were sent to the president and General Mattis. And it's because it's easy to grow the plants and it's easy to do the extractions. The most famous use of ricin was by the KGB and their assassination of this dissident, Georgy Markov, in London. One of the KGB officers came up and had a few hundred micrograms of ricin on the tip of an umbrella and just stabbed him in the leg and then ran away. And he died 4 days later from the ricin poisoning. A couple hundred micrograms of this compound. The reason why ricin is so toxic is because it disables translation permanently and catalytically. And this is the reason why it has such potency in organisms. And we're going to talk about how that is in the upcoming slides. I wanted to begin the lecture on translation by talking a few things about the genetic code, which I believe were touched on in the original introductory central dogma lecture. First is that the conversion, translation of nucleic acid sequence at the RNA level to amino acids at the protein level is done 3 bases at a time, so-called triplet code. The table, the codon table, is shown on the right-hand side. It happens to be blue, but you don't have to memorize the table. I don't have the table memorized. But there's a couple salient features that I want to emphasize here. First, just some math. We have 4 bases. It's read 3 at a time. So there's 4 times 4 times 4 possible combinations of 3 base sequences that comprise codons. This is how you have 64 combinations on the right-hand side. And I already slipped up by scooping myself. Each one of those 3 bases we call a codon because of this triplet code. And you'll see mechanistically why the reading occurs 3 bases at a time. A few more terms and characteristics that you need to know. first, continuing with the numbers, we have 20 canonical amino acids, and yet there are 64 codons. And so what that implies is that there must be some degree of degeneracy in the relationship between codons and amino acids. And there is, you can see it in the table. Multiple codons code for the same amino acid. And so this is how you get the funneling from 64 down to 20, plus a couple of additional ones that I'll speak about in a moment. Even though there's the degeneracy, the assignment of a codon to an amino acid is unambiguous, meaning that every time one sees, a ribosome sees U, it will encode phenylalanine. It's not, there isn't any bifurcations or uncertainty in the code when it's read 3 at a time. And then the third characteristic is that codons are read in a non-overlapping manner. What this creates is what we call a reading frame for the gene and the protein product that occurs 3 bases at a time. So the ribosome will read a triplet codon, encode an amino acid, and then step forward 3 bases to the next codon to encode the next amino acid. These were big mysteries 50, 60 years ago to figure out exactly how the information in ribonucleic acids was translated into proteins, because one could imagine any of a number of ways in which the information could be encoded. And the last thing is that even though that there are 64 possible combinations, there are only 61 that encode for amino acids. 3 are what we call stop codons that do not encode for amino acid information, but encode for where the translational machinery should stop translating and release the final polypeptide. Those 3 stops are shown up here on the top. Oh, and then the very last thing, which I don't have a bullet point for, but you do need to know, is how translation starts. There's only one start amino acid, one start codon, AUG, that encodes for methionine. All proteins start by translating a methionine or a modified methionine. As with other facets of the central dogma, we're going to begin with an overview and then dive into progressively more and more details. And we will also begin with prokaryotic translation, and then where it makes sense, we'll do a compare and contrast with eukaryotic translation. First, the broad strokes. Here we have the mRNA template that will be used for translation. These are the 2 subunits of the ribosome, more to come on those, but we have a large subunit and a small subunit. And then there are these transfer RNAs that act as the interface between the nucleic acid information and the amino acids that are going to be conjugated to one another to form the polypeptide chain. During initiation, the first codon, the start codon here, is recognized by the tRNA and, along with additional sequence information, prompts the assembly of the ribosome around the mRNA at the translational start position. From there, the ribosome moves along the mRNA template, 3 bases at a time, 1 codon after a time, and at each point, a new tRNA comes in that is complementary to the information encoded in the mRNA, and then the polypeptide is transferred over 1 amino acid at a time to the last tRNA that got incorporated into the ribosome. At each one of these cycles, the ribosome moves ahead 3 bases, 1 codon, continuing to elongate the polypeptide chain until at the very end, a stop codon is encountered, and rather than a tRNA going in, a protein called a release factor enters into the ribosome, and then rather than transferring another amino acid, transfers water to the growing polypeptide chain. Polypeptide is released, ribosome falls off the mRNA, and the mRNA remains for another round of protein translation. Yes? Can tRNAs get reused? If you are referring to yes. So the actual tRNA itself, we're going to talk about charging and uncharging of tRNAs with the amino acids. And so the answer is yes. I think I'm going to talk about that right now, in fact. Yeah, so next topic is the tRNAs themselves. This is a 2D projection of the tRNA. There's a lot of hairpin character to the tRNA's 5' end, 3' end. And there are a couple of key features here. First is this anticodon. The anticodon has a degree of complementarity to the codon on the mRNA species. I'll talk more about how that complementarity is not as strict as it is for nucleic acid recognition. And then the other site here is at the 3' end, which is where the amino acid that will go onto the growing polypeptide chain is covalently coupled to the tRNA via this ester bond. So a tRNA in the earlier cartoon that is not loaded with an amino acid or not charged with an amino acid will look like this. And then once it's been added to the appropriate amino acid, it will look like this. So these are the ones that will go into the ribosome and be conjugated during translation. So anticodon, this stem here that will accept the amino acids called the acceptor stem. And here's a 3D display of what tRNAs look like. So it's not just a cross-like structure there. It is much more 3D character, but the features are the same: acceptor stem, anticodon, and then all of the rest here is basically to put provide the right orientation for recognition by the ribosome and these accessory factors that I'll talk about. So this is the tRNA. First, you need to envision this tRNA coming in and sampling whether or not it's a good match as an anticodon to the codon on the mRNA. The rules of base pairing and anti-parallel character of how nucleic acids come together still hold. So we have the 5' end here going to the 3' end. So in this cartoon, this must be the 3' end, and this must be the 5' end of the template. So on this topic of complementarity, here's where things get a little bit more interesting related with respect to the types of base pairing that we discussed with DNA, RNA, and DNA. Even though there are 61 codons that encode for amino acids, there are not 61 tRNAs for each one of those codon sequences that I showed previously in the table. Part of the reason why is that there is a position in the anticodon, which is the so-called wobble position. This wobble position is in the first position of the anticodon, again going 5' to 3', and it's in the third position of the codon, again 5' to 3'. This has to do with some of the chemical modifications that occur to tRNAs. It also is a function of the geometry of, you saw how that tRNA was structured. So it's not undergoing a perfect helical, right-handed helical conformation like what we do with DNA. And so because of that, the rules of hydrogen bonding are a bit more flexible. Some of these base pair the way that you would expect, even in the wobble position. So A's and C's are pretty strict, but if you have U or G in the anticodon, you can see that there's some flexibility. And in addition, there's a chemically modified base called inosine. We've seen this before. That exists in anticodons that provides a good degree of flexibility in what position of the codon can be tolerated. A little bit more about inosine. As I said previously, this arises from a deamination event of adenine. So you take away this amine here, and then you're left with inosine after a couple of rearrangements. And this is favored in certain tRNAs because of the way that they're structured, like I had shown previously. Lots of flexibility in the base pair. And here's an example how a single tRNA whose anticodon is IAU can accommodate codons. Right? Again, so this is always 5' to 3', so IAU, which goes U, A, and then either U or C or A. Any of these codons in an mRNA sequence will give rise to an isoleucine amino acid. Yes. Yeah, and some of the things I'm going to add today, every once in a while, a book chapter illustration flips it around and violates that rule because it's trying to emphasize the base pairing. And so I'm going to make a point to remind when the 5' to 3' is flipped around just for clarity. How do the tRNAs at that acceptor stem get conjugated to the amino acid? This is an enzyme catalyzed event. It is catalyzed by a set of enzymes called amino-acyl tRNA transferases or amino-acyl tRNA synthetases. Mechanistically, what they do is shown here schematically on the right-hand side. They have two substrates, one of which is going to be the unloaded or uncharged tRNA, and the second is the free amino acid that ultimately gets conjugated to charge or load the tRNA. Each amino acid that's going to get translated has a dedicated transferase to undergo the chemical conjugation that I'm going to talk about. And you can rationalize this because you know what the chemical structures of the amino acids look like. They're all different. There's going to need to be some type of binding pocket on that synthetase to be able to use it as a substrate, and this is the reason why you have that specificity. So what occurs? So the synthetase will bind a free amino acid as well as ATP, and the first step is to hydrolyze ATP between the alpha and beta phosphates, release a pyrophosphate. Okay, so that's a high energy bond that's being released, and then conjugate AMP to the amino acid. This moves the amino acid into a high energy state. That then when the synthetase recognizes an unloaded tRNA, it releases AMP, and now will create that ester bond between the amino acid and the tRNA, creating a charged, loaded, activated amino acid. The key result of this enzyme catalyzed reaction is that loaded or charged tRNA has a high energy bond, that ester bond that links the amino acid to the tRNA is a high energy state. And that high energy state is exploited by the ribosome during translation to elongate the polypeptide chain. So right now we've hydrolyzed an ATP to raise the potential energy of the delta G of the tRNA, and then that's going to get cashed in during translation. That's going to cover the elements of base pairing, codon and anticodon base pairing, as well as the loading of tRNAs for translation. What I'd like to do next is circle back to mRNA and the features of mRNA from a ribosome's point of view, so to speak. There's a couple terms in here that I'm going to try to say you don't need to know because I've never seen them used in practice, but there are some terms that are important to know. What we have here is prokaryotic mRNA and then a eukaryotic mRNA on the bottom. We're going to focus on the eukaryotic mRNA, capped on the 5' end, polyadenylated on the 3' end, and then the RNA itself inside. Not all of the RNA will code for protein. there's an upstream portion that is not translated, called the 5' untranslated region, or 5' UTR. That 5' UTR is a nucleic acid sequence. It can impact the ability of the gene to be translated, but the sequence itself is not translated. And the reason why it's not translated is the ribosome has not encountered an AUG yet. There hasn't been a start codon yet. And so it's sequence that impinges on translation but is not immediately translated. On the other end of the sequence, you have a 3' untranslated region, or 3' UTR, and this is everything after a stop codon has been encountered. So the stop is not right at the very, very end where polyadenylation occurs. There can be very long UTRs at the end, and to a first approximation, the 3' untranslated regions play a role in the stability of the RNA. So there are hairpins or other consensus sequences in there that have an impact on the overall stability of the RNA itself. So there's important control elements there, but are not immediately impacting translation. In between is where translation occurs. And in between is the coding sequence, or CDS, of a gene. So 5' UTR, coding sequence, 3' UTR. The only reason I keep this cartoon on here is I will touch on one unique characteristic of prokaryotes that does not hold for eukaryotes, and that is a dedicated ribosome binding site that provides instructions of where the ribosome should start. And more to come on that, I think, in the next slide or two. First, a little bit more about ribosomes. You got a brief introduction on them, I think, in a pre-lecture at the very beginning of the class. We'll speak more about them in structural detail today. I already mentioned that they have two subunits, large and small. More information on the largeness and the smallness of them soon. But I show the ribbon diagram here on the right-hand side of the ribosome to emphasize two things. One, ribosomes are comprised of RNA and protein, the color coding here for the protein and the RNA. And that when you look at it crystallographically, I hope that you can appreciate it, that the vast majority of the ribosomes is RNA. The protein are appendages on the outside. They serve some regulatory roles, some to aid the structure of the RNA, but the ribosome is largely RNA. Question? Would the ribosome be considered a SNRP? Yeah, and so so SNRPs, so small ribonucleic particles, this is not small. These are very big. And so it is a ribonucleic particle, if you will, but much larger than the SNRPs like that we talked about in splicing. In this diagram, large subunit is on the left-hand side, small subunit is on the right-hand side, and then envision the mRNA going in between those two subunits. The bacterial ribosome, large and small, are called 50S and 30S. You put the two together, regrettably, you don't get 80S, you get 70S. More to talk about of the S in a moment. In eukaryotes, we also have large and small, but it's a 60S and a 40S, also not adding up to 100S, it adds up to 80S. This begs the question, what the heck is all this stuff about S? And if I tell you it stands for Svedberg units, that tells you nothing, but I just give it to you for entertainment purposes. It's this guy called Svedberg who came up with a unit of measure for how readily large particles sediment during ultracentrifugation. Ribosomes are so large that if you have them in a tube and you spin long enough and hard enough, you can pellet them. And depending upon the size of these macromolecular particles, they will sediment faster or slower or at different speeds. And so this S, the Svedberg unit designation, is to give an indication of how long and hard you have to spin. Big complexes get larger S units because they pellet easier. Yes? It's purely gravity-driven sedimentation. So it's a rough indication of the size. And the reason why historically is we've had ultracentrifuges for a long time, so you would simply spin, take off the supernatant, spin harder, take off the supernatant, spin harder. And so it was a series of ultracentrifugation steps, and then whatever you got at the bottom of the tube each time got that designation. It's a bit of a historical artifact, but that's what we have. Here's the cartoon form of the ribosome, akin to what I showed, rotated 90 degrees in the previous slide. There are a couple important sites of the ribosome that you need to know. I already talked about where the mRNA goes in. The mRNA binding site, okay, is here at the interface between the large and the small subunits. And then there are 3 dedicated sites that play key roles in the cycle of translation. There's the A site, and A stands for activated tRNA. This is where the charged tRNA goes in. It's right here. Activated site, amino acyl site. Remember it however you want to do it, but it's the A site. In the middle of the ribosome is the P site, which is where the polypeptide chain resides ordinarily, peptidyl site. And then after the transfer of the amino acid has occurred, or the polypeptide chain has occurred to the incoming tRNA, the discharged tRNA resides in the E site or the exit site. This is where the unloaded tRNAs leave from the ribosome. And as you'll see, the sequence of events are charged tRNAs going into the A site, receiving the polypeptide chain from the P site, the ribosome moving, and then expelling the uncharged tRNA out of the E site, and then repeating the cycle until the stop codon is encountered. So we'll talk about how this process gets started first. Starting translation is called translational initiation. And here's one of the places where we have a bit of a divergence with prokaryotes and eukaryotes, but also some commonality. Both prokaryotic translation and eukaryotic translation involve proteins called initiation factors, or IFs. And what these do is work together with the subunits of the ribosome, as well as the tRNAs, to identify where the translational start is on an mRNA. In prokaryotes, there is a dedicated ribosome binding site. It's called a Shine-Delgarno sequence that at the sequence level instructs where the ribosome should assemble, guided by these initiation factors, hydrolysis of GTP, as well as the docking and incorporation of the initiating tRNA, which in prokaryotes is a modified form of methionine called Formyl-methionine. You remember FMLP, that PAMP, and we said how Formyl-methionine was really important for prokaryotes to be able to translate protein. This is the reason why. They can only start translation with a formulated methionine, and that's why the innate immune system exploited it and evolved the PRR for it. So translational initiation in prokaryotes involves the assembly of these initiation factors, formulated methionine tRNA, small and large subunits. And then once you have this formulated methionine into the start position, the ribosome assembled, now you're ready to begin translational elongation. There are a few wrinkles in eukaryotes. First, we don't use formulated methionine, we just use regular methionine, although AUG is the same as the codon. The details on the initiation factors are different, so the naming scheme is different. We call them eukaryotic initiation factors. So the details vary, but they do need initiating factors. And then the third difference is that there isn't a Shine-Delgarno sequence. More what appears to happen is that these eukaryotic initiation factors with a methionine-charged tRNA exploit the 5' cap, the modification on the mRNAs that we talked about before, and then you have the small subunit binding and a kind kind of scanning that occurs in the 5' region of the mRNA until a start codon occurs. You get GTP hydrolysis of the initiation factor, assembly of the large subunit of the ribosome, and now you're competent to begin translation. Why don't we use Formyl-methionine, and why do bacteria do? Starts to get to be more of a philosophical question. I can explain why you might have two different, vastly different organisms set up to be this way. The amino acid has to, the modification there structurally is giving some type of instructions to the ribosome about the start site. And even though ribosomes are deeply conserved through evolution, there are divergences between eukaryotic ribosomes and prokaryotic ribosomes. And so if you'll allow me to wager a speculation, I think that the pairing there has to do with the coupling of how translational initiation differs in prokaryotes relative to eukaryotes. And it may be some combination of that Shine-Gilgarno sequence, the formylated methionine, and the structure of the prokaryotic ribosome to work together to help define translational initiation. Whereas by contrast in eukaryotes, it's a little bit slopperier, if you will, about exactly where everything starts. And so the rules may be slightly different about how you do the start. There's also sometimes evolutionary events that happen just by random chance. And then they have such a profound impact, they just say canalized throughout evolution. So it's hard to say exactly for sure. That's initiation. This is a really complicated diagram. I'm going to break it down in a couple of slides in a moment. But what these slides deal with is the process of translational elongation. This is the cycle that occurs over and over again each time a new amino acid is incorporated into the polypeptide chain. So we're going to start here on the top, work our way around this cycle, and then we're going to end up back at the top for another round of amino acid incorporation. The broad stroke steps of translational elongation is the recognition of an appropriate tRNA that's bound by the ribosome in the A site. The second is the formation of the peptide bond, the peptide backbone that makes the amino acids. So that chemistry between the A and the P sites is the next step. Then after that, you have a translocation of the ribosome forward 3 bases, one codon. And then the last step that's coupled with that translocation is the expulsion of the discharged tRNA from the E site. And this goes again and again, but more details in the next several slides. Blow up of that figure on the right. For the first cycle around, let's just assume that we have a formylated methionine or a regular methionine right at the beginning of translational initiation. It exists in the P site here. So this is also where the polypeptide chain would occupy after we've gone around one cycle. So the first event here is binding of an appropriate charged tRNA into the A site, which is this step here. This step is guided by an elongation factor, or an EF, called EFTU. Is it? No, okay. EFTU binds to GTP, so GTP ACE, and it helps guide or assess the recognition of the complementarity between the charged tRNA that's coming in and the mRNA sequence that's bound by the ribosome. There's some sampling that occurs with this EFTU, GTP, and charged tRNA complex going into that A site. If it doesn't have good complementarity, it just gets kicked out. However, if there is enough base pairing, if it does conform to that codon table that I showed from before, this charged tRNA, EFTU, GTP complex will persist in that A site. You can even trap it crystallographically. So here's EFTU. Here is the tRNA that's being presented by TU, and then the P and the E sites by way of reference. If that amino acid charged tRNA is the appropriate match for the mRNA sequence, then what occurs is the hydrolysis of those two GTPs to GDP, so release two inorganic phosphates, and now EFTU and the two bound to the two GDPs dissociates from the complex, dissociates from the ribosome, and that stably puts the amino acyl tRNA into the A site. And we are here. The next step is extension of the peptide chain. This part is catalyzed by the ribosome. And so I might have said this before, but the ribosome is also a ribozyme, right? So it's RNA, all that structured RNA is helping catalyze a chemical reaction, and that chemical reaction is the formation of the peptide, the peptide bond. So within the ribosome, using the high energy bond, that ester bond that I talked about before, there's a transfer of, in this first cycle, around the methionine to the next amino acid over here that just entered in to the A site. That's thermodynamically favorable because of that high energy ester bond. Now you've elongated. After the peptide bond has formed, there's recognition by another elongation factor called EFG. I apologize for these names. I don't know what the history is on them. They're sort of awful, but they are what they are. It's another GTP binding protein. It will bind to ribosomes after the peptidyl transfer has already occurred. And that as it binds here to the ribosome, I need to go back. There's a translocation of the ribosome, 3 bases ahead, 1 codon ahead. Now the polypeptide chain is back into the P site. EFG hydrolyzes the GTP to GDP and PI. The whole thing falls apart. And the unloaded tRNA is expelled out from the E site. Now we're back to the top of the cycle, except now we have 2 amino acids rather than 1. And this process of translation is occurring from the N terminus, the methionine, the start methionine, to the C terminus. This is the reason why we have the convention of N to C terminus that Professor Barker harped on in the very beginning of class. And that's why it couples to 5' to 3' with nucleic acid sequence, because this is the direction in which translation occurs. It's the direction in which nucleic acid synthesis occurs. Here's a cartoon form of it. Ribosome A, P, E. EFTU, sampling, presenting tRNAs to the A site. There's a match. GTP hydrolysis, release of EFTU. Peptidyl transfer from the P site to the A site. Binding of the EFG. Hydrolysis, okay, it's not really a wagon wheel, but translocation of the ribosome. Now what was once in the A site is in the P site. What was once in the P site is now in the E site. Discharge tRNA exits. Repeat. Here's everything in words. I think I just said it in words, but so that everything is in writing, you have it here as study guide. Now let's talk about how do you stop this roller coaster of translation. The way that we stop translation, translational termination, is through those stop codons that were in the table, those 3. Stop codons have a unique chemistry that occurs when they're encountered by the ribosome. When the stop codon is in the A site, right, so now you have the whole polypeptide chain, everything is done with translation, but remember that the polypeptide is still covalently bound by the ester bond to the last tRNA that was bound and the last amino acid that was translated. So how do we finish? When the stop codon is encountered, rather than another tRNA going in, what goes into the A site is a protein, or a family of proteins called release factors. So It's not a tRNA, it's a protein. And what it does is it occupies the A site, and then during what would ordinarily be a peptidyl transfer, water is transferred, hydrolyzes the ester bond, and then creates the carboxyl terminus of the protein and releases the polypeptide. Upon release of the polypeptide, the whole translational complex falls apart. Small and large subunit dissociate, and the polypeptide diffuses away. What's really neat is that if you look at the structure of the release factor, shown here, it's pretty similar to a tRNA. So it's like molecular mimicry between what a protein to look like a tRNA to provide a trigger for translational termination. Okay, so I'm going to repeat your question just so that I make sure that I understand it. You had said, we talked about the carboxyl terminus, and you generate that C terminus by this water transfer to the ester bond. And your point is that there are carboxylated groups all throughout the peptide sequence, including the peptide chain in the middle. Those are the ones you're referring to, not to carboxylic acids on the side chains of amino acids. In the bond, right? Okay, so they participate. They're comprising that ester bond in the charged state. But I think what you're asking is, what is the chemical reaction that the ribosome is catalyzing? Rather than the formation of a carboxylic acid, it's creating a bond between a carboxylic acid and an amine to form an amide. So it's forming an amide bond. That's the peptide bond, rather than a carboxylic acid at the end. And that's the magical catalysis that the ribosome does. Yeah, so your question is, circling back to some of the elements of transcription, in eukaryotes, they have the cap-binding proteins on the 5' end and poly-A-binding proteins on the 3' end, and they could join up with one another and circularize the transcript. How does that relate to all of this if you have initiation and termination happening at the start and the stop codons? You still have that happening at the start and the stop codons, but if you think in the crowded environment of the cell, if you have a ribosome that has docked, done the forward search on the 5' end, starts translation, goes all the way around to the other end, and then falls off. Even when it falls off, it's still in the vicinity. There's still a high local concentration of large and small subunits there, much higher than if they had to diffuse away from elsewhere in the cell. Thus, it's still kinetically favored for those large and small subunits to find the 5' end again of the same site, even if they've fallen off before they got all the way around to the end, because they fall off at the stop site. So it's still a local enrichment, but it's not running a complete loop like you probably envisioned when we talked about it during transcription. Very good. We have a few bits and pieces of things to add in here. One, in lecture one, we talked about proteins and amino acids and folds, alpha helices, beta sheets. How do those come into play here? When protein folding happens, it happens in real time with translation. As those polypeptide chains are getting elongated, the solvent exposed, the water exposed parts that have been translated up to this point are folding into their thermodynamically favorable state as it's growing. Okay, and so that this is a key determinant to getting a stably folded protein, is to have it fold as the translation is occurring. Second is the energetics of this. We do more of this energetic accounting when we do metabolism, but each time we have to synthesize an amino acid, there is an energetic cost to this. We needed 1 ATP to load up the tRNA in the first place, create that high energy ester bond, and then 3 GTPs needed to be hydrolyzed, 2 from EFTU and 1 from EFG. This is the reason, if anything, this creates a motivation for all of that transcriptional regulation that I talked about in the preceding lecture. There's a cost to making proteins. So if we can regulate genes so that they're only made on demand when it's advantageous for them to be there, that saves energy for the cell. Third thing is something I don't believe that we've touched on before, but that translation, it ties in a little bit with the circularization question from before. Is how do you rev up the efficiency of this whole process when you have an mRNA species? And one of the ways is that you have multiple translations occurring at the same time on an mRNA species. In the cartoons on the slides from before, I showed one ribosome marching along. But in reality, you don't have one ribosome on one mRNA. You have multiple ribosomes at different stages of elongation along the mRNA template. That assembly of mRNA and a whole bunch of ribosomes is called a polysome, or polyribosome. Here's an electron micrograph of one of these polysomes. You can't see the mRNA template, but it threads through here. The very dark, larger, electron-dense things that you see in the image, those are the large and small subunits. And the squigglier parts here, that's the elongating polypeptide chain. So 5' of the mRNA is here, translating N to C terminus as it goes along. And you can see the longer polypeptide chain off down at the end. And then the last thing is about translation is I wanted to circle back to this concept of operons in prokaryotes. In the operon model, you have one mRNA encoding multiple protein products, multiple genes. How does that work from the perspective of translation? Well, what occurs is that one mRNA species will have multiple Shine-Delgarno sequences or other flags to mark where translation should occur inside the mRNA species. And so a polycistronic mRNA is an mRNA that encodes for multiple protein products like an operon. And the way that is accomplished is by having multiple ribosome binding sequences upstream of each one of those proteins that needs to be translated. And those can occur simultaneously, akin to this, but now you're having it occur at different protein products. Now that we have our heads around translation, I wanted to go back to some of the things I mentioned delicately related to DNA damage and VDJ recombination, and this relates to changes in our DNA and how they could impact protein function. And so changes in our DNA could occur by mutation, deamination, ionizing radiation, collapsed replication forks, or they could just occur by variation in the population. You don't look like me, different, right? And so there's diversity in our DNA sequences, and there is diversity in our coding sequences. It's much rarer than in our non-coding sequences, but we do have differences in the proteins that we express. And so we can put that into the context, we can put those DNA changes in the setting of translation, and then they fall into very clearly defined categories of what those mutations, we'll call them mutations are, but they could be variants as well. And what they relate to are changes in the nucleic acid sequence that have or do not have an impact on the amino acid that is encoded by that change. So first we'll talk about missense mutations. Missense mutations are a change in the DNA sequence that gives rise to a change in the RNA sequence. Here's one of the ones where the DNA, they're showing the template strand, 3' to 5'. So T goes to A, T goes to A, C goes to G. This is in the proper orientation, and GAA is a codon for glutamate. If you change the middle base from a T to an A, now you have GUA, and it encodes a different amino acid. So what you have is a substitution in that amino acid sequence from a glutamate to a valine. And if you'll recall, retention, glutamate is very acidic, hydrophilic. Valine hydrophobic. Okay, so this could impact some of the thermodynamics of the protein, how it folds, or it may have no impact. Depends. So those are missense mutations. Nonsense mutations are the same change, but what the substitution is that occurs is rather than with another amino acid, a stop codon is created. For example, leucine here, codon is only one base different from a stop codon, and so if the substitution occurs there in that middle position, now you've stopped translation prematurely. And the third category of substitution is what's called a silent mutation. And silent mutations come to bear on those wobble positions. Remember, there's some flexibility in what could base pair in that third position of certain codons. There you could conceivably do a change. Here's one example for proline, GGG. If the GGG goes to a GGT, this is the wobble position. You still make proline. You've changed the nucleic acid sequence, but the amino acid sequence is the same.
So if you inherited single cell from both your parents, would that still be a missense mutation, or would that be non-mutation because you inherited?
Yeah, your question is, what about inherited mutations like the sickle cell allele or other ones that we'll talk about? There the terminology changes. We will usually call them missense variants, nonsense variants, to get across the idea that it's an inherited variation rather than a, do we talk about somatic and germline differences? Yeah, so germline variants, you usually call as variants, where you have somatic mutations, so things that occur as a result of the environment or stresses or other types of events in the soma. That's where we will often refer to them. That is where we'll refer to them as mutations. So missense, nonsense, silent for substitutions. There's another category of both variation and mutation that we've also touched on, and these are insertions or deletions. I need to tread cautiously on insertion and deletions because it makes no sense if we don't have an understanding of this thing called a reading frame. Translation occurs 3 bases at a time along the codons. Envision that there's a DNA event. This could occur by variation, errors in splice sites, VDJ recombination. This is also where you'll see that imprecision of joining. If there's an insertion or a deletion of a base, and if that insertion or deletion is not a multiple of 3, you have changed the reading frame, the 3-base reading frame of that gene from the perspective of the ribosome. We'll walk through this example here. Let's say that this is the starting sequence. Presume it's a start methionine, and it should ordinarily go lysine, phenylalanine, aspartate. 3 bases at a time. If there is an insertion mutation that puts a G into the DNA, that's going to be encoded as a C. Translation starts as a methionine, but now by virtue of the altered reading frame because of the insertion, the next amino acid is a glutamine, then a valine, and when you shift the reading frame, almost invariably what will occur is that the ribosome will encounter a premature stop codon off the reading frame, off the original reading frame. And so the net result usually is a variable degree of truncation, I've used that term before, shortening of the translated polyprotein product as a result of insertions or deletions. This illustration is an insertion, but you can go through the same thought exercise with a deletion, and the same type of frameshift premature stop codons will be encountered. So we're going to keep to time. We'll leave this as a discussion point afterwards if you want to talk about it on Piazza. Think about among those categories of mutations, frameshift, nonsense, silent, missense, which of these do you think would confer new functions or have the potential to confer new functions to a protein? These are so-called gain-of-function mutations. And what I mean by that is enabling a protein to do something that it doesn't ordinarily do. And which would be loss of function, meaning taking something that a protein that would ordinarily do and then removing that function from the mutated protein product. You can make some inferences about which of these you think would fall into one category or another. All right, we have all the tools now. We need to get back to Ricin. I have to introduce a couple of topics that are a little bit ahead of their time, meaning you haven't seen them yet in lecture, but bear with me. Some of the things, though, should be old hat. We've seen them in other lectures previously in the class. So the ricin is a toxin, crystal structure as shown here. Like other toxins that you have seen before, think botulinum toxin. It is a multi-subunit protein. It specifically is a dimer of two different proteins, a heterodimer of two proteins called Ricin A and Ricin B. Ricin A is an enzyme, and we'll talk about the chemical reaction it catalyzes soon. That's the cytotoxin, but it cannot enter into cells on its own. To achieve cell entry, it uses the other subunit, Ricin B. Ricin B is a lectin. We talked about lectins, right? What do lectins do? Wait, I can't remember. Did I tell you what lectins do? Yeah, okay. It's not exam time yet. Or that was an exam before. Lectins bind sugars. Okay, this is the lectin-mediated pathway, looking at things like lipopolysaccharide or things like that on bacterial surfaces. Lectins are a broad category of proteins. And our cells, as well as every other cell type, has a bunch of sugar-modified proteins on our cell surfaces. After the exam, we'll speak about sugar modifications on proteins, but for now what you need to know is that we have a whole sugar coating on our cells. And so the Ricin B as a lectin, as a protein that binds sugar-modified proteins, that acts as a docking site for this toxin to bind onto the cell membrane. Also, by virtue of its ability to bind multiple sugars, it has this property of what's called an agglutinin. And what agglutinins are multivalent proteins that can bring other proteins together and cause them to aggregate and fall out of solution. So it not only does it bind sugars, it can bind a lot of sugars and bring things together with high avidity. Ricin A and Ricin B are joined together covalently, but reversibly by a disulfide bond. Think back to botulinum toxin and the different chains and how the subunits are brought together. The path of ricin, this is, if you were stabbed with an umbrella or you ingested it, okay, it's got to get inside the cells to do damage. So I want to speak briefly on the cell entry, largely to motivate protein trafficking, which we'll cover after the exam, because the ricin path in many ways is the reverse of some of the later steps of protein trafficking. We have the alpha and beta subunits outside of the cell. Sugar-modified proteins on the cell surface are called glycoproteins. They're also glycolipids, sugar-modified lipids. The Ricin B binds both of them, so it just sticks to the cell surface. By virtue of sticking on the cell surface, that creates a trigger for internalization of the toxin. More on that when we do protein trafficking. And then as it gets inside of the cell, it goes through a normal endocytic pathway, which you've seen in other settings before. So into the endosomes. Some go back, some get degraded through lysosomes. We touched on these briefly, but a small fraction will go backwards from the normal way that proteins are secreted. And more on this after the exam, but if you go back from secretory vesicles, the next step back in time is the Golgi apparatus, which you heard nothing about up to this point, but as a brief prelude, this is where a lot of sugar modifications occur, and there's trafficking that can occur bidirectionally among these different vesicles. It's the stuff that gets back to the Golgi that's bad from the cellular perspective. This is where escape of the alpha and beta subunits of the ricin toxin occur, and now you have them into the cytoplasm. Once they're in the cytoplasm, reducing environment, disulfide bond is reduced, and now you have free Ricin A. Now we're on step 7. And I already scooped myself here. All right, so reducing environment, disulfide bond falling apart into the cytoplasm, exactly like botulinum toxin from before. Now that there's free Ricin A in the cytoplasm, I mentioned that it was an enzyme. The enzymatic reaction that it catalyzes is a depurination event. It's a depurinase, if you will. But its substrate is the ribosome. All right, so where this depurination occurs is on ribosomal RNA, takes off a purine, and then as a result of that, the large subunit of the ribosome can no longer bind those elongation factors. All right, so all of those protein ribosome interactions with EFG, EFTU, or the eukaryotic analogs to them no longer can occur. So you've broken that cycle of elongation, and translation stops. And the reason why you have this profound lethality of ricin is that you need very little of the ricin to do a lot of damage to the translational machinery in cells. Because it's an enzyme, it will undergo numerous rounds of catalysis. Right? It's not consumed by that depurination event and deactivate many ribosomes in the cell. So here's the rough estimates. 1 molecule to 50,000 ribosomes. And mammalian cells have about 10 million or so, 1 million to 10 million ribosomes as a rough estimate. So you don't need that many molecules of ricin to get into a cell to completely stop translation. No translation, no cell viability. All right, a couple more words on ricin. There are actually uses for it and its impact on translation aside from bioterror. We use it in, well, I don't use it, but it is used in the lab a lot if you're a translation aficionado. You can use it to freeze the steps of translation at key steps if it's used judiciously. It's been important for several of the crystal structures that we've shown along the way in this slide. And it's also a really nice example of protein nucleic acid recognition. Recall that the ribosome is mostly RNA, but the ricin toxin is a protein. And so to get a deeper understanding of RNA protein binding, which is quite complicated, it's used as an exemplar. And then the last thing to touch on for this is there are potentially therapeutic applications of ricin, specifically in cancers because of its potency. So they are looking into things called immunotoxins. We've talked about antibodies before. Imagine there is a cancer that has a massive surface expression of a protein on the plasma membrane. Okay? And you can use that as a marker for the cancer. What people are looking into is you raise an antibody that binds very tightly to that surface protein and then modify that protein so that it's covalently coupled to ricin. Now the antibody provides the honing to get to the cells that have that high surface expression of the molecule, and then if that antibody complex is internalized, and can in principle go the same pathway as ricin, and you only need to get a couple of those molecules into a cancer cell to be able to kill it. It's still in experimental phases, but I checked this afternoon and people are still publishing papers on it, so it's still an active area of research. Right. If you're interested in translation and microbiology, immunology, and cancer biology, up on 7th floor of PIN here is our ribosome person at UVA. And probably should have left more time for discussion, but I have plenty of time for questions. Yes?
What are some treatments of ricin
Yeah, the treatments for ricin, waiting and crossing your fingers, because it's not, there's no intervention. mostly it's palliative. You just have to let it run its course. It's like some of the other biotoxins. You just hope that the enzymes kick out after a while, and hope that the individual can see it through. I think I've read that also children are especially susceptible for it. So anybody like with castor plants and accidentally eating the beans, is a major concern. That's why anybody who's growing the plants are supposed to make sure that they don't they don't germinate or form seeds or whatever for safety reasons. Yes? Yes. Yeah, so the question is, at the Golgi, how do you have this membrane escape? Yeah, and so to answer this with what you know now, at each point, the toxin is in the lumen of a vesicle, but the environment of the lumen of the vesicle is changing. And so the pH is changing, the enzymes that are going along with in each one of those vesicles is changing. And what occurs is that as it gets all the way back to the Golgi, there's a conformational change because of the microenvironmental change in the lumen there that enables you to pass through a membrane. So I actually don't 100% know the details if it's a transient pore forming, but there's some examples of some of these toxins undergoing a conformational change to form an alpha helical structure that will poke a hole in the membrane and allow it to leach out. But this is, it's tied to the environment of the lumen of the vesicle in which the toxin is reside. It's the same theme that occurs with viruses, other toxins, botulinum, same thing. It's finding the right microenvironment where you can punch a hole in the membrane to escape. Oh, okay, so related question, how does it get out of the lysosome? It doesn't get out of the lysosome. That's the degraded pathway. There's wastefulness to this, but it's a numbers game. All you need is enough to serve an evolutionary advantage to dispel predators from eating your seeds. All you need to have is enough to get into the cytosol to be a deterrent. Same thing also happens with other pathogens and things. It's a numbers game. It's never we draw it as 100% cartoon, but a lot fall by the wayside before that. But enough gets in to be able to continue on. Yes? I heard everything except for the most important part, which was the last part. Oh, okay, yes. All right, so your question is, there are methionines in the inside of proteins. Why doesn't translation start there? Or do I have that right? Or in a different reading frame. Great, yeah, and if you play around with sequences and things, so if I take a gene and I just start searching for reading frame, so-called open reading frames that could translate, the true open reading frame is rarely the very first ATG that's encountered in the sequence. It's just when you find that ATG, you see a much longer sequence until you hit a stop codon. A couple answers to that. So that first one. The thinking is that some of this may happen, is that you may go and it starts to translate a little bit, right? But it hits a stop codon so quickly that it just kind of quickly goes and falls apart and then goes back on and keeps going on from there. So there is a little bit of efficiency up on that first starting point, and to the extent that that is coordinated by those initiation factors, in other words, is there some sort of sampling to figure out when it terminates prematurely, it's usually quite fast to hit that premature stop. But there is evidence of some leakiness that occur with the ribosome in making these smaller products. They're just hard to see and hard to evaluate. But if you have one that gives rise to a long, elongated protein product, the ribosome is going to dwell much longer in translating on those, and so the net result is the bulk of the translation is dedicated to the longest or open reading frame of the gene. Your second question is thematically similar, but the answer is different. If you have a methionine inside the cell, there can be this phenomenon called leaky ribosome scanning, whereas it starts at the 5' site, and then it just overlooks the first start methionine, and then keeps going, and then finds another methionine in frame, and then translates the other part. That happens, I wouldn't say all the time, but there are many examples I can think of where what you then create is a short product and a long product, both of which share the same C terminus, but they have different N termini. Or the longer form is going to have an extended N terminus that's absent from the shorter form. So that definitely happens. I've encountered multiple examples in my lab where we've gotten thrown by what the protein we're looking at because it's translated at 2 different points, and we see 2 bands on a blot or things like that. So there is some imprecision with translation related to where it starts, not where it finishes. The release factors are quite good in that sense.
Is that why you get multiple genes or one transcript in prokaryotes, but maybe not
It is a mechanism of protein diversification in mammals, or excuse me, in eukaryotes, that you won't have in prokaryotes because there they require that Shine-Dalgarno sequence, and unless that's internal into a translated product, you won't see it. I think it's simply a different way. I think it's like an evolutionary divergence. The advantage of the operons is, and a polycistrons, is that if you have a process where the stoichiometry is really important, it's great, because then one gene gives you one copy of each one, and they're all getting translated at the same time. So it's a really nice way to turn on a whole pathway with the right balance. And if you have them all as independent mRNAs, they each have to be transcriptionally regulated. They each have to be translated separately. And so there's the potential for less coordination, but then there's also the potential for more fine-tuning, if you will. So each one of them can adapt and have its own transcriptional regulatory circuit controlling how the mRNAs are controlled.
Is the actual synthesis of ribosomes like the translation?
Okay, so the synthesis of ribosomes in the nucleolus, how does that compare to translation in the cytoplasm? So in the nucleolus, this is where the rRNA is made. So it's really a transcription process more than a translation process. The protein parts of the ribosome are translated just like I showed before. And my understanding is that they get loaded on after the ribosome, all the RNA is being translated and assembled. So there's a secondary assembly of those protein subunits of the ribosome concurrent or after the transcription of the ribosomal RNA. So my understanding is that it's really the RNA-centered part occurring in the nucleolus. There's also evidence of ribosomal heterogeneity, in that even within a cell, you have 10 million ribosomes. They're not all identical. Some may have some of those protein subunits. Others may have different sets of subunits, and they may have different translational efficiencies to different mRNA targets. That's a pretty new area. And then was maybe one last question?
Yeah, so you mentioned how peptide chains with termination transfer through water. Can you elaborate on that? Does that actually mean water? Can you just elaborate on that process?
The transferring of the water with release factor to the polypeptide chain, which, okay, so I don't have a good cartoon for this, but we talked about that high energy ester bond with all the rest of the polypeptide. So it's polypeptide O, and then the rest of the tRNA on that acceptor stem. Usually that's the site for where the amine as a nucleophile will come in and form the amide bond. But rather than that, you just have a water molecule, the electrons on the water molecule attack the O, and now you get OH. And so what it's doing is breaking that ester bond, and then rather than forming a peptide bond, an amide bond, you simply create a carboxylic acid there, because there's no amine in the water.
So like the water
It's a hydrolysis, yes. Mm-hmm. All right, excellent. Good questions in the last 15 minutes. I'll see you on Tuesday.